disjointed things today. But the first thing I wanted to talk about is the bottom problem here, which is one of my favorite problems. It's a classic, probably 20 years old. I almost ran into discovered the paper that this was originally done in, but I actually think they faked a lot of this data. Okay, so we're looking at, we've already covered group 1. We've covered group 2. Okay, that problem, that first problem, 1113, is about group 3. Okay, but this problem is about group 4. And it's about what is the effect of insulin on group 4. Okay, so this is the insulin responsive glucose transporter. Okay, and this problem used to have probably more impact maybe 10, 15 years ago, and I'll explain why after we solve the problem. Okay. So they're looking at, in this experiment, at adipose cells, even though we've already noticed that besides adipose cells that carry the group 4 skeletal muscles, somewhere that skeletal muscles take up about 80% of the blood sugar. But that's because there's so much of it, of skeletal muscle relative to the amount of brain or the amount of liver, okay? But they take up the glucose in response to insulin. Okay, so this problem starts out with the recognition that in the presence of insulin, the adipose cells take up more glucose than in the absence of insulin. Okay, they also start out with understanding that there's a group 4 transporter on the adipose tissue, in the adipose tissue, not group 1 or group 2. Okay, somewhere. So they did this. So they added insulin to this cell to see how is glucose uptake going on. They used some protein synthesis inhibitors, okay, so that the cell could not make more GLUT4 transporters. And they discovered that insulin had no, or that these protein synthesis inhibitors had no effect on insulin's activity. Which, what does that suggest? If you could add protein synthesis inhibitors and insulin still has an effect on the cell, then insulin is not stimulating what? Protein synthesis inhibitors have no effect. So in other words, you get the same response whether proteins are being synthesized or proteins are not being synthesized. This is, by the way, a standard experimental strategy. So insulin is not stimulating the production of more glucose transporters. Okay? So when you see an experiment that's done in the presence of a protein synthesis inhibitor, one of the question that they're trying to rule out is, do we need to make, is this hormone responsible for making more glucose transporters? And their answer clearly is no. Okay? They also did the same with glycosylation inhibitors. So they wondered, does insulin alter the glycosylation pattern on the glucose 4 transporters? And the answer is no, because whether you have the carbohydrate there on the surface or not, it doesn't. It's on the group 4, and that doesn't matter. Okay. So now they have to look at some other strategies. So we know that insulin's not stimulating the synthesis of glucose transporters. It's working with the glucose transporters that are already present in the cell. It's altering the activity of those proteins already present in the cell. So they do 2 experiments, okay? One of them is just poorly reconstructed here, so I'm sorry about that. So you're gonna have to guess. It's really hard to get the information out that they're asking from this teeny weeny curve, but you're stuck with it. Okay, so one of the questions is, when we compare adipose cells that have been treated with insulin and adipose cells that have not been exposed to insulin, here is the really the Michaelis-Menten kinetics. I'm going to try to blow it up, and I'm not sure I can do it justice. Okay. Okay, let's go with that I drew on the board. Okay. And the question, one of the important questions is Vmax altered in the presence of insulin? Yeah. Because here it is, the Vmax is low in the absence of insulin, Vmax is high in the presence of insulin. Okay, so this changes. Does the Km change? I see some people shaking their heads yes and some people shaking their heads no. What do you think?
Yes.
Okay, let's take a look. Let's take a look at how well I drew this. Okay, so this is supposed to be Vmax for, does that look all right to you? Okay, so here's Vmax for no insulin, half Vmax, be about there. I'll use green. Okay, there's Km, okay? So you have to take a look at, compare the Vmax for no insulin to find the Km for the group 4 in the presence of insulin. Okay, let's try the Vmax. Very good. Okay, that's about half. Is that it? Okay, I'm gonna go with that. That might be a little bit high. Okay, and there's that would be Km. So, depending on how you draw it, not really. Okay, and it really, the difference in Km is just because of how I drew it. The difference in, if you find a difference in Km, it's really a matter of how they drew it. But the reality is, and the conclusion, okay? In order to compare Kms, you really have to calculate, you just can't eyeball it. You really have to calculate what is the Km for this curve, what is the Km for this curve. Okay, so that, and you can try it, but it's with that teeny weeny figure, it's really, and you don't even know where K, or you don't even know where Vmax is. So we'll say, not really. Maybe a little bit. So in other words, it transporters, and this is what you're supposed to come, the conclusion that you need to come to, is that insulin doesn't really change the affinity of the transporter for glucose. That's what you were supposed to recognize, but the drawing is so poor, it's extremely difficult to come to that conclusion. Now that's a surprising conclusion that probably at one time in history would have thrown students off, because probably that group of students learned in their physiology class that insulin increases the transporter's affinity for glucose. That used to be taught at Simpson all of the time, because that was the kind of the conventional wisdom about the effect of insulin on the uptake of glucose in adipose tissue and skeletal tissue. So it should have come as a surprise to you that no, the affinity doesn't change. Insulin doesn't have a change, does not change the affinity. Okay? So that was, well, okie doke, that's an interesting thing. Okay, so then they tried to find where is glucose, the GLUT4 protein located in the cell. So they used a molecule, acetylcholasin. I thought it was a lectin, but probably, I don't think it is. This is a molecule that binds to GLUT4, and when it binds to GLUT4, it inhibits GLUT4, and it really doesn't matter in this condition, because all they're doing is binding acetylcholasin to GLUT4 to find where it is. Okay, in order to find acetylcholasin, okay, it was given a radioactive isotope, tritium. Okay, so they used the tritium label on the acetylcholasin to monitor where is the acetylcholasin binding. Okay, and the table 11.1 shows you the results. Okay, so in the absence of insulin, where's the GLUT4? In the absence of insulin, where's GLUT4? Yeah, it looks like it's predominantly, there's some on the plasma membrane, but most of it's on internal organs. Organelles. Let's say internal membranes. Okay, when you add insulin, the results should suggest what about the location? It's now located where? Yeah, predominantly on the plasma membrane. So what accounts then for the increase in Vmax?
For the plasma membrane.
Yeah, so the total number, or higher concentration or a higher total number of GLUT4 receptors on the plasma membrane. In the presence of insulin, fewer GLUT4 transporters on the plasma membrane in the absence of insulin. And some of you wrote that in your sample writing. And somebody wrote that that internal membrane. Somebody wrote that the internal membrane is the Golgi apparatus. It's really not. I don't care what your physiology book says. It's really not. There is a storage vesicle that holds the GLUT4. And the storage vesicle sometimes moves GLUT4 back and forth between the Golgi and the storage vesicle. Okay, but insulin triggers the movement of GLUT from the storage vesicle to the cell surface. It's not coming from the Golgi. So it used to come from the Golgi at one point when it was being made. That's where insulin went through the Golgi, but it's predominantly stored in vesicles. Okay, we probably won't cover the exact pathway that leads from insulin binding to the adipose cell to the release of GLUT4 or to the movement of GLUT4 to the plasma membrane. Okay, but this was clearly different from what most students learned probably 15 years ago or so. So that's why this experiment was important to be added to the textbook. Okay. By the way, are there any questions about what we did yesterday in this particular problem? And has anybody solved 1113? Anyone take a minute at that? I would suggest you do that. I'll give you a clue. You cannot calculate the rate, and you cannot calculate Vmax. What you're calculating is the ratio of the rate to the Vmax. So you're really calculating what, how close to Vmax is this transporter operating. Okay, it says it's operating at 50% Vmax, is it operating at 75% Vmax? So what you're calculating is actually how close to Vmax is the transporter operating. Okay. So give that a try. I would put mine in a table. Okay, so you can, it probably is best in the table, and then you can compare GLUT1, or GLUT3 in this case, and GLUT2. So one of the things that I would have said several days ago, but get on to another topic, is that in general, these transporters are substrate specific. In other words, the glucose transporters could, under certain circumstances, transport other monosaccharides, but they're really best for transporting glucose. I'm going to give you some Km. Here we go. Mark my place because I want to come back to. Oh, but. So we're looking at GLUT1. For the D form of glucose, which is the glucose you make when you digest polysaccharides, or the glucose you make if you're doing gluconeogenesis, it's always D glucose, has a Km of 1.5 millimolar. Okay. The lactose, I'm going to put D-galactose, because all of these are going to be used. Is an isomer of glucose, so it's a monosaccharide. Many of you eat galactose probably every day. And what foods contain galactose? It's rarely found as a monosaccharide. It's in a disaccharide form.
Milk.
Huh?
Milk.
Milk, yeah, part of lactose. Yep. Okay. It is probably not being picked up by GLUT1 because the Km is 20 millimolar. We probably have to get some other thing. Let's see, mannose. You know what mannose is? I was thinking maltose. Mannose, another isomer of glucose. 30. Oops, this is 30. This doesn't matter. Okay. In other words, galactose and Dartwell better find another transporter. L-galactose, so if you manufacture, if you make the L form of glucose, its Km is way over 3,000. Close. So that's kind of, and that, I think that's a general thing you can say about lots of transporters are extremely specific for the ligands that they transport. Okay, how many amino acids are there? 20. Do you suppose you can have 20 different transporters, one for each amino acid? No, because I wouldn't ask the question. But can you imagine? Your cell membrane would have to be enormous in order to have adequate uptake of amino acids. So the amino acids actually, the amino acid transporters carry more than one amino acid. And what they transport really is divided into different classes. So transporters, I'm going to talk about one transporter that carries 7 amino acids, and they all have similar properties. Okay, so I'm going to write out the names of the amino acids that are used by this particular transporter, and see if you can guess. Okay. See if you can guess what the properties are. Okay. Anybody remember what these 3 amino acids have in common? They're all of they're the aromatic amino acids, right? Okay. How about isoleucine, thymine, and I'm missing one, methionine. Okay, can you think about any common feature? Don't worry so much about methionine. How about the leucine, isoleucine, and valine? Huh? They're not polar. They're all carbon hydrogen. And methionine falls under that category too, because even though methionine has a sulfur group in it, the sulfur is not very electronegative. It's essentially not polar. Okay. So there's a single transporter that carries these 7 amino acids. They're the 7 bulky hydrophobic amino acids. Okay, so we didn't put glycine on that list, which the R group is just a hydrogen. We didn't put alanine on that list. Because alanine's R group is a methyl group. So these are the larger hydrophobic amino acids. Okay. So people with who are born with, you were tested actually for this disease, PKU. Phenylketonuria. You don't remember being tested for that, but and probably your parents didn't know, but your foot was, somebody poked stabbed your foot, drew a little bit of blood, and determined whether or not you had phenylketonuria. Okay, people with phenylketonuria can't break down phenylalanine. So you find phenylalanine in the proteins of mom's milk or formula. And it sort of stays that way. And normally, what you would do to break it down is to convert phenylketonuria into tyrosine. No, hydroxyphenylalanine. And the last tyrosine. Yes. Okay. So the phenylalanine level in the blood increases a lot. In fact, if it's untreated, people with phenylketonuria will have 20 times more phenylalanine in their blood than normal, than you would normally find. What's going to happen to the uptake of the other 6 amino acids? Yeah. If you overwhelm this transporter with 20 times too much phenylalanine, then the tryptophan and the tyrosine and the leucine and all the other ones aren't also going to be taken up. They're going to be excluded. So there's a lot of competitive inhibition here, or that too much phenylalanine will compete and the other amino acids can't get in. Okay? These are all important for protein synthesis, but is there one that might be particularly important for protein synthesis? Huh? I've heard it. Methionine. How come? Yeah. So methionine happens to be the first amino acid of all proteins. So the start codon AUG codes for methionine. Okay? So when this is happening, okay, when there's too much phenylalanine, there's really not enough methionine getting into the cell, and that even slows down. So proteins that need to be made are not going to get made. You can't initiate protein synthesis if you don't have enough methionine. So the brain is not so actually in the fetus, the mom is breaking down the phenylketonuria for the baby. So the baby gets a normal amount of phenylalanine, but after birth, when the baby no longer has mom's enzymes to break down phenylalanine, then that's when the phenylalanine levels start to rise. So that's why they measure it in you. It's not a good fetal test because you can't tell. Okay. What would you do if somebody had phenol? Why did they test you at birth? What would they do to you or for you at birth? Well, first of all, what do you suppose happens to people with phenylketonuria if they're not treated? You don't know? Huh? They don't grow their, and they also turn out to be really hyperactive because you're not making enough tyrosine. And lightly pigmented because melatonin is made from, or melatonin is made from tyrosine. They're generally mentally retarded. They have a low IQ. But with treatment, you can achieve the effect on brain development. Because the brain still develops quite a bit after birth. Okay? But if your brain's not capable of making the required proteins, the brain is going to stop developing at the time of birth. So what's the best way to treat then people with PKU?
Inhibit phenylalanine production.
Can they give you something to break it?
Well, it's in the diet, it's in your food, it's in mom's milk, it's in formula.
Will it inhibit uptake then?
But then you inhibit the uptake of all the other guys too.
Can they give you something to break it down?
Huh?
Can they give you something like artificial to break it down?
No.
Could you get some of the enzymes from the mom to help degrade it?
Oh, no, because generally it works, it might be working in the mom, but once you get into the baby, you can't really add the enzyme. That doesn't work because the enzyme has to be inside of cells, and you have to talk about how do you get it in the cells. What's currently done is you take the baby off formula and off mom's milk, and you put the baby on a special diet that lacks or has very, very low phenylalanine. So phenylalanine is an essential amino acid, so you can't remove it from the diet, but you can lower it. You can reduce the amount of phenylalanine. So that's why you were tested. If you had turned out that you had phenylketonuria, they would have pulled you off mom's milk or formula or whatever you were drinking. They would have put you on a special diet. Yeah.
Do you have to maintain that diet then for the rest of your life? Or
Well, that's a great question, Kelly. The answer to that question is no. Okay, anybody want to make any stabs on why not? Why when you get to be, why aren't people with PKU eating normal diets by the time they're 20?
The brain's done developing?
Huh?
The brain's done developing?
Well, that might be, but that's partly true. Okay, good, that's fine. Something else that makes it extremely extremely difficult to get drugs up into the brain. The blood-brain barrier is not developed in infants, so the brain sees a 20-fold increase in phenylalanine at birth until the blood-brain barrier is established, and then once that blood-brain barrier has developed, the blood-brain barrier controls how much phenylalanine will reach the neurons. How do you know? You know, and so part of the problem is, so the kids are on a special diet. Apparently it's not fun. And they go to a birthday party, they're 5 years old, they go to a birthday party, and there's great food. Now, if you're a grandma like me, I would give in, say, oh sure, here, have this, have a hamburger, have a hot dog, what the heck. So it turns out it's a horrible diet. It's hard to keep on the diet, so people gradually go off the diet. Any other, is there any other way you can do this, treat people with phenylketonuria besides remove the phenylalanine?
Increase the other stuff.
Increase the others, yeah. If you can increase, this is what I did research on this, increase the other the 6 amino acids, you can reduce the phenotype of phenylketonuria. You can block the mental retardation and the hyperactivity and all that other kind of stuff. I did this in mice, and I discovered that I put in as much tryptophan as I put in leucine, isoleucine, all the other amino acids. I kept killing the mice. They would die. They would die in a half hour. And I finally died. All right. So I started modifying my ratio, and I took out reduced the tryptophan, and they started to stay alive. I guess we were just making too much serotonin. So anyway, yeah, there's probably alternative treatments to phenylketonuria. Adding excess amount of the other 6 amino acids works, but you got to watch out for the tryptophan. You can tryptophan yourself to that. That was every, I mean, I went for months just killing mice. It was so discouraging. Okay, that takes care of antiporters. Okay, so that takes care of passive transporters, and we're going to all of a sudden bounce into active transporters.